Hey friends, I'm sad to say that my voice is still a little bit off here. I've got a bad cold and I'm all sniffly and nasty and unpleasant here, but I thought I would go ahead and try and do this. So last week I made a video critiquing Simon Dan's approach to a creationist, and I made the point that he got several things wrong about the evolution of vision. So I thought what I would do is today I'd say a few words about the evolution of vision and explain a little bit about the background to this. I'm struggling. I'll make my voice work, but we'll see. Anyway, so what do I want to say? Well, today what I want to talk about just is something very, very basic, and that is the evolution of photoreception. Okay, this is distinct from vision. That what we're going to be discussing is how certain cells have acquired the ability to respond to light. Okay, that's it. We're not going to be making images. We're not going to be using this for vision. These are capabilities that we can use just to detect whether it's light or dark out. That's all it does. And this is probably the oldest of the sensory capabilities we have. It's pretty basic stuff. So let's take a look at how that works. Okay. So the core of this is a ubiquitous, really common sort of molecule. This is retinal. Retinal is a vitamin D derivative, and it's got some really useful properties for vision in that it's got this long chain. You see that right there, that chain, that can absorb a photon. So if you shine light on it, it will unkink, it will straighten out. Pretty basic, huh? It's just absorbing a little energy from a photon. And then there's an enzyme that uses ATP that can put the bend back in it. So we've got a sensor now. You can just have this molecule sitting in your cell and measure the concentration of the cis form versus the trans form, and you have a measure of the amount of illumination falling on the cell. Amazing. Now this is really old stuff. We're talking a molecule that became useful 3.5 billion, 4 billion years ago. So this is really ancient. It's been around for a long, long time. Another interesting detail about this retinal is that basically what it's doing is all by itself it's converting a photon into a conformation change. And it's a conformation change that requires energy to undo. So there's energy going into the system to cause this change in shape. And there's a hypothesis going around that, hey, you know, this could have been an easy way to get some energy. It's not very efficient. You don't get a lot of energy out of it. But you can imagine that early in evolution, cells had retinal, and they could use it to bask in the sun and generate a little bit of energy that they could then use in metabolic processes. And this is something that would have preceded the evolution of chlorophyll. Chlorophyll is a much bigger, much more complex molecule, but it's also much better at converting sunlight into energy. But it's entirely possible this is around 3.5 billion years or so ago, and that cells were using it to do a crude form of photosynthesis. One of the cool things about that is retinal has a different color spectrum than chlorophyll. Chlorophyll, as I'm sure you know, is a molecule that absorbs in the red and the blue wavelengths and reflects in the green. And that's why our trees all look green. Well, not my trees. My trees all look white and naked. But yeah, if you're in a warmer climate, trees and grass are green because they got chlorophyll and it reflects the green light. Retinal, on the other hand, would instead absorb green light. That's just a property of its chemical spectrum, and would reflect red and blue light, so it appears purple. So one interesting hypothesis is that there was a stage in the early evolution of life, they call it the purple Earth hypothesis, where if you came upon planet Earth and looked down upon it, it wouldn't be a big green ball with swaths of blue where we had oceans. Instead, it would be very purple, which would be cool. Okay. So anyway, retinals, it's essential. It's common. You have cells loaded with retinal, not just in your eyes, but everywhere. Okay, there's another step to this, though, that we have to consider. So we've got retinal, which undergoes this conformational change in the presence of light, and retinal boosts the performance of this molecule. This is a protein. This is opsin. And retinal is embedded. Let's see if I can see that kind of pink thing right there? That's the retinal molecule inside of the barrel of the opsin molecule. And this acts as an amplifier. So when retinal changes its shape, it butts up against the walls of the barrel that it's in and causes a conformational change in the opsin protein, and then opsin does interesting things. It's then activated and can do all kinds of stuff to expand the response to that light signal. So yeah, opsins are everywhere as well. You've got loads of opsins in your eyes. Another feature of opsin, of course, is that it allows for spectral tuning. So some forms of opsin allow long wavelengths to pass through and activate retinal. Others allow short wavelengths. So by using the right opsin, you can get color sensitivity in your retinal. Okay. So good so far? So retinal undergoes a conformational change, triggers a related conformational change in opsin, and then opsin is going to activate a G protein. Yeah, if you don't know about G proteins, G proteins are kind of awesome. G proteins are small proteins that are embedded on the interface of the cytoplasm of the cellular membrane, and what they do is they act as shuttles. So here what you can see, there's our G protein in kind of a teal color there, and over here we've got a G opsin linked receptor. So opsin or G protein linked receptors are a particular class of receptor that when they bind something, what they do is they activate a G protein. And then the G protein shuttles around in the membrane and can go activate other enzymes. So this is another component of vision, is you got retinal that activates opsin, and opsin then activates a G protein, and the G protein then wanders around in the membrane, bumping into other proteins, and can thereby activate enzymes. Are you with me so far? Sounds easy, right? Like I said, this is ancient stuff. This has been around for a long, long time in evolutionary history. Now, what about in light detection? Again, it's the same thing. So you've got the opsin, that's this blue thing right here. Light shines on it, and then it activates a G protein right there. And the G protein then activates this molecule over here. This is cyclic GMP phosphodiesterase. PDE for short, okay? So we're going to activate an enzyme here. And that enzyme has the function of breaking down cyclic GMP, converting it to 5' GMP. Yeah, that's really exciting, isn't it? The point of that is that if you shine light on this system, it turns on PDE, which then breaks down cyclic GMP, so the cyclic GMP concentration declines. It drops down radically. And that has the effect that in the membrane of this cell, right there, there's an ion channel. This is a cyclic GMP gated channel. So it allows sodium to pass through. So if you drop the levels of cyclic GMP, this channel is not going to be activated anymore. It's going to close, which means sodium can't pass through the membrane. And what that represents is a reduction in current. So when you shine light on a photoreceptor, what it actually does is it doesn't get the photoreceptor excited, it depresses it. So it's going to have less activity. Kind of counterintuitive, but it works. It's the same. As far as the neural systems are concerned, you just have to respond to either a increase or decrease in current. So it can recognize this. Another feature of this is, what I've talked about so far, you've got opsin, which is embedded in a membrane, and you've got G proteins that are embedded in a membrane, and here you have phosphodiesterase, which is embedded in a membrane, which is activating a cyclic GMP gated channel, which is embedded in a membrane. Everything's going on in the membrane. So when you look at photoreceptor cells, what you tend to find is, like you see right here, so there's a rod, and it's got all these infoldings, all these complicated discs of membrane in there, because you want to increase the membrane area. The more membrane you got, the more sensitive the tissue is. So that's why you've got this peculiar structure. It's just full of membrane, and all the membranes have all these proteins embedded in them to do the work. So far, so good. Now I got to add a complication. Oh, you're saying a complication now? Oh, what was all this stuff before? Yeah, this is a complicated cascade, but it's not that complicated, right? Any of you who've taken cell biology, biochemistry know that biology is really messy that way. Here's the complication, is that what I've been describing so far is this cell here. This is a photoreceptor, diagrammatically, in a vertebrate, for instance. And this is called a ciliary photoreceptor because it's modified cilia to make this thing. And in this diagram up here at the top, there's the opsin. There's the G protein that's going to be activated. There's the phosphodiesterase. Cyclic GMP is going to be reduced by that, and we're going to get a reduction in current. So that's what we see down here, is if you shine a light on that ciliary photoreceptor, you get a drop in the current. It declines rapidly. Okay, so far so good. That's not the only way you can do things. And in fact, what you find is that there's another class of photoreceptor over here. This is a rhabdomeric photoreceptor. And as you might guess from the name, rhabdomeres. What are rhabdomeres? Rhabdomeres are the individual subunits of an insect eye, for instance. Okay, so insects have compound eyes. Each of those little facets is part of a rhabdome. So we got a rhabdomere here. If you go to an insect and poke into its eyeballs and take a look what it's got, there you can see, hey, it's got an opsin. That's familiar. It activates a G protein. It is a slightly different G protein than the one activated in vertebrates. And again, that's okay. There's a huge number of different kinds of G proteins. And in insects, what happens is that G protein then activates this enzyme. That's phospholipase C, which is a very different enzyme. Phospholipase C, what it then does is that it breaks down phosphoinositol phosphate into inositol phosphate. Yes, a twist. Very exciting. Okay, so we're going to break that down. And inositol phosphate then binds to a membrane channel and opens it. So, like I said, it doesn't matter. Polarity can be interpreted any way you want. But in this case, if you look at an insect, what will happen is activating that particular rhabdomeric receptor causes an increase in excitation. So that's what you're seeing here in this little graph down here. You flash a bit of light at the eye, and it gets a spike in current. that then gradually declines. Oh, is this stunning stuff? Does this mean that these are radically different photoreceptors and they follow different evolutionary paths, and that, hey, yeah, these are not of a common origin? No, it's not. Doesn't mean that at all. You will not be surprised to learn that evolution is perfectly comfortable with this, because what we've got here, to summarize, is we got very similar pathways. You got the ciliary and rhabdomeric receptors that all have opsins, that are all stimulated by photons striking the retinal in the opsins. And then they activate a G protein, and the G protein then activates enzymes. It's just different enzymes in the two kinds of cells. So we start to diverge there in this point, okay, but it's still phototransduction. In vertebrates, we're going to reduce the concentration of cyclic GMP. In insects and invertebrates, we're going to produce IP3, and that's going to affect diacylglyceride. Yeah, don't worry about the acronyms. In vertebrates, then, we're going to close sodium channels, which is going to cause the current to decrease. And we're going to hyperpolarize the membrane. And down here you see in insects and other invertebrates, it will open these transient receptor potential channels, and that will cause a current increase, which will then depolarize the cell membrane. Yeah. Don't you love this stuff? Yeah, a little biochemistry, a little evolutionary biology, it all fits together. Now, like I said, maybe this is terrible news. Maybe that means that these are divergent enough that they don't have a common origin. But no, they do. We can look in more detail at these cells here. And here's a very revealing example. This is Planoneris. This is an annelid worm. And when you look at this lovely creature, what you can see is, okay, over here, you see this? That dark blue region? That one there? Those are eyes. And those eyes are for giving the poor worm vision. And those use rhabdomeric photoreceptors. Okay, so it's got a battery of rhabdomeric photoreceptors in there that it can use to sense the world around it. What are these things over here? These little brown things. Those also have photoreceptors, but has ciliary photoreceptors. So I was misleading earlier when I characterized, okay, ciliary photoreceptors are vertebrate, rhabdomeric are invertebrate. No, that's not the way it works. They have them both. So invertebrates have both ciliary and rhabdomeric photoreceptors. And so it's just that the difference is they use the rhabdomeric photoreceptors to sense the world around them, to generate vision, to see things. They use the ciliary photoreceptors for circadian rhythms. So they want to be able to sense the time of day, basically. And lots of organisms do this. Lots of them care about just programming in their little internal clock to tell what time it is, what time of day it is. Because, for instance, you might be a nocturnal animals. You don't want to go out during the day, so you get inactive and you sleep during the day, and then later your internal clock tells you wake up, get out there and do things. It's the same with us. We also have these circadian rhythms where they tend to wake you up at particular times of the day for most of us. For instance, me, I wake up promptly every morning at 6:30. Can't help it. It's my internal clock. It's just ticking away in there. Okay, so Planoneris has both kinds. It has rhabdomeric for sight and ciliary for its internal clock. What about me? What do I do? Well, it turns out that we also have those ciliary receptors in our eyes, and we use those to see things, for vision. But we also have a population of rhabdomeric photoreceptors in our retinas, deep in the ganglion cell layer. And those aren't used to, they're non-visual photoreceptors. All they're there for is to tell you what time of day it is. So we got those ticking away in us. So basically we're the same. Invertebrates and vertebrates, they have both kinds. You have ciliary and rhabdomeric eyes. But they've reversed how they use them. So invertebrates use the rhabdomeric for vision, the ciliary for circadian rhythms. Vertebrates use the rhabdomeric for circadian rhythms and the ciliary for vision. But yeah, we've got them both. That sort of suggests that the last common ancestor of invertebrates and vertebrates had both kinds of eyes, both kinds of photoreceptors in their heads. Okay, this is a complicated diagram. I'll try to keep it short and simple here. What this is the phylogeny of opsins. So what they've done is they sequenced all these opsins and then categorized them by similarity. So what you can see is that there are three major branches of the opsins. So up here at the top, this one is those rhabdomeric opsins. So all these animals have rhabdomeric photoreceptors. Down here are the ciliary opsins. So all these animals have ciliary opsins. And one thing you might notice is, for instance, if we just look for people, there's Homo, that's us. We have that ciliary opsin, which is unsurprising because that's what we use for vision. That's what our eyes are working with. But then you follow this branch up here, and let's see if I can find. Oh, there it is. Look at there, Homo encephalopsin. There are a bunch of opsins in us that are not used for vision, but are instead used for sensing light levels. For example, we actually have an opsin that's embedded in our skin cells. Okay, lots of cells have opsins. They're sitting there, they're recording light levels, and those are used, for instance, for melanogenesis. Yeah, you go out in the sun a lot. Shine the sun. Have you ever wondered why when you're out in the sun you don't have a systemic level response, but if I go out with my clothes like this, I would get this part tanned and this part would stay pasty white like a good Minnesotan back here in my arm. That's because it's locally sensed by opsin molecules in all of your skin cells, which I think is kind of cool. But a lot of these are going to be the rhabdomeric opsins and not just the ciliary opsins, which are mainly up here in my eyeballs. Okay, we can map these all out. Now, I mentioned last time that I really prefer molecular data to fossil data. That fossils are easy to visualize. It's easy to imagine how a fossil tells you something about evolutionary history. Molecules are harder, but I think they're better. You get higher resolution, and also the other key thing about them is lots of things don't fossilize. Eyes don't fossilize. These molecules don't fossilize. But what you can do is reconstruct the phylogeny by comparing different species and seeing how the molecules sort out in this kind of phylogenetic tree, which is kind of powerful. So yeah, we can look at these opsins. We can see, yeah, there was a history that there were different kinds of opsins that were playing different kinds of roles in our evolutionary history. So that's what we're seeing here. I mentioned that there were three big branches. We won't worry about it too much. But here what you can see is, here are the protostomes. Those are things like insects and other invertebrates. And they have ciliary and rhabdomeric opsins. Yay, that's what I just told you. Over here are the deuterostomes. We are deuterostomes, so that's things like vertebrates and echinoderms and other such things. Those are all deuterostomes. And we also have a ciliary and rhabdomeric opsin and photoreceptors in here. And again, don't worry about the RGRs. I'm trying to simplify as much as I can. Anyway, so what we can see here is, if we trace this back to the eumetazoa, the true multicellular animals, they're back in here, and they would have had two kinds of photoreceptors right from the very beginning. And then in the bilaterians, that again is lots of different organisms. That's us, that's insects, that's spiders, that's squid, all these things. The bilaterian animals, the ones that are bilaterally symmetric, they have produced a third one, this RGR, but don't worry about it. But we can also see that all of their descendants have both ciliary and rhabdomeric eyes. And the cnidarians, again, we won't worry about those, but yeah, there's another branch. Nematostella and cnidops are really interesting animals. They just have two instead of the three that we do. But the point being that we share this evolutionary history, and we all have all of these photoreceptors. They've just specialized in various degrees in different ways in different organisms. Okay, this is actually a shortened version of a presentation I give in my classes for my intro biology courses, so freshmen can grasp this stuff, right? So you'll have no problem. And so I've got a little summary here. These are the key points I would want my students to take away from this. And the first one is that all animals use a common core of retinal plus opsin for vision. So we all use that. That's how we see, is retinal plus opsin. Then we got this process of transduction, where that's all that business of taking opsin that's been activated by a photon and then stimulating a G protein and then an enzyme and changing channel properties, etc. It's complicated. And it's really kind of a Rube Goldberg machine. It's just a series of steps that are dictated by history more than just efficiency. That we could design a much more efficient system, I imagine. But that's not how evolution works. It's kind of cobbled this together over time. But interestingly, different species vary more in later steps than in the early steps in transduction. So they've all got opsin, they've all got retinal, and then they got the G proteins, and then they vary later with different enzymes that they activate, which then switch on yet again different channel proteins in the membrane. And again, that fits with what we'd expect from evolution, that you start with a common pattern and evolution generates gradual divergence into new patterns. Okay, little detail here. So vertebrates use ciliary photoreceptors for vision. There's the pathway that works over. And invertebrates use rhabdomeric photoreceptors for vision. And there's that slightly different pattern here, where invertebrates we're going to activate phosphodiesterase and reduce current. In insects, we're going to use phospholipase C, and we're going to increase current across the membrane. Makes perfect sense. It's arbitrary which way you go. We picked one path, invertebrates picked another one. And then the last common ancestor of vertebrates and invertebrates had both rhabdomeric and ciliary receptors. So there's a unity there. Even though we're using different ones for vision, we've got the same molecular history in the formation of vision. And we, in particular, use ciliary receptors for vision and rhabdomeric receptors for circadian rhythms. And in invertebrate, we just flip that around. Use different receptors for those things. And that the important point there is, again, why are molecules better than fossils? Because we have detailed molecular descriptions of the evolutionary history of the eye. We do not have detailed fossil descriptions. Eyes just don't preserve very well. And the key differences are those things like ciliary versus rhabdomeric, and you typically can't see those in a fossil. You definitely can't see which kind of opsin they're using, or which kind of G protein they're using. That's lost for us forever. But by comparing different phyla, we can actually reconstruct the common characteristics of our last common ancestor. All right. So I'm going to leave it at that. That's enough to digest for a little while, right? But I just want to point out, I'm thinking next week I'll talk a little bit more about the actual evolution of the eye structure. Again, keep that distinction in mind. Photoreception is one thing. All you need is cells with particular molecules in there, and we can use those to sense the presence of light. Eyes, on the other hand, are a major advance on that, because now what you're going to do is you're going to take an array of these sensors, an array of photoreceptors, and you're going to use the structure of the eye to focus an image on that array so that we can actually sense shapes and patterns and colors and all that kind of cool stuff in the object we're looking at. And I'll just close this by mentioning, yeah, Darwin noticed this. This is from the Origin. He says, suppose that the eye, with all its inimitable contrivances for adjusting the focus to different distances, for admitting different amounts of light, and for the correction of spherical and chromatic aberration, all functions of an eye, could have been formed by natural selection, seems, I freely confess, absurd in the highest degree. I really like this quote because Darwin is trolling future generations of creationists. He really is. This is one of the most frequently cited passages from the Origin by creationists, because they say, oh, they just love this part. Seems, I freely confess, absurd. They never go to the next page. It's amazing that it just tells you something about the creationist mindset, that, yeah, you get this quote and then you stop cold and you say, oh, it's absurd, we're done. But then if you actually read the Origin, you go on to the next page, what you find is it goes on for several pages where he describes all these intermediates, these different stages in the formation of an eye. And, for instance, you got an optic nerve, it's surrounded by pigment cells, it's covered by translucent skin. You got aggregates of pigment cells, apparently serving as organs of vision without any nerves. We'll talk about some examples of that. And that to make in an eye, all you need is a depression in a layer of these photoreceptor pigments. And that gives you some directionality. But yeah, I'll save that for next week. I've talked long enough. All right, I'll let you go there. Thanks for stopping by. I'm going to go rest my voice now because, man, that was hard. Talk to y'all later.